In Chapter 4, we discussed all of the elements leading to protein structure. We discussed primary sequence, secondary structure, tertiary structure, quaternary structure, all of which are elements that build upon the previous element to give you that native three-dimensional structure that is hopefully going to lead to function for a given protein. Now, in Chapter 5, we're going to turn our attention to that function itself. Largely, what we're going to focus on here is how a protein or enzyme interacts with its ligand, its substrate. We're not going to yet talk about the catalytic output that comes later. Really, this chapter is focused entirely on how a protein or enzyme interacts with its ligand. And so you'll see we will discuss this through a number of different perspectives, which include structure, also include more functionally centered studies that invoke some math. We will talk about some math along the way. But what I really want you to just pay attention to here is the underlying theme is a description of how an enzyme or protein interacts with its ligand. Much of our efforts here are going to be centered on looking at hemoglobin and myoglobin. These are very well characterized examples of proteins that bind a ligand. Myoglobin was the first protein whose structure was determined on an atomic resolution level, so for that reason we know a lot about myoglobin. It has been very, very well characterized, and hemoglobin is probably the next best characterized protein, where hemoglobin is simply a tetramer of myoglobin-like polypeptides. So myoglobin is the monomer, and if you take four polypeptides that look a lot like myoglobin, they are not exactly the same, but look a lot like myoglobin, and put them together, you have hemoglobin. So the commonality here is that myoglobin and hemoglobin are both oxygen binding proteins, and for that reason we can take myoglobin as a starting point to build into the complexities that you will see appear later for hemoglobin. So we're basically going to start simple and then build into a more complex description. So in both cases, remember, they are oxygen binding proteins. So to give you a little bit of the physiology, hemoglobin is going to bind oxygen in the lungs. It is then going to take that oxygen, move it through the blood to muscle tissues where it is going to dissociate the oxygen, and that oxygen is not yet where it needs to go. In order to do aerobic respiration, you have to take that oxygen a little bit further to get it into mitochondria where it will be needed. So the protein that's going to do that is myoglobin. So hemoglobin takes oxygen through the blood to the muscle, lets go of it, and myoglobin picks it up and carries it to finalize that delivery. I view the myoglobin hemoglobin system kind of like the FedEx truck almost, where you have a distribution center. You take your packages ultimately to the local destination where they're going to be, and then another truck picks it up and takes it to your porch. That's what is basically happening here. Hemoglobin is your mass transit. You were taking packages wherever, to the local destination, and then myoglobin is going to finish it off, take it to your doorstep where it is needed. Now, let's go ahead and jump into myoglobin. Let's start where I said we would. Let's start with the structure of myoglobin. I have shown it to you before, but now we're actually going to walk through it and talk about some of the characteristics of myoglobin. So first, myoglobin, it is a single polypeptide chain. It is monomeric. It is 153 amino acids in length from N-terminus to C-terminus. It is also an all-alpha helical protein. If you look at the structure here, you'll see a bunch of cylinders. So these cylinders they are another way to represent your alpha helix. And all of your cylinders are connected via linkers. So this is an all-alpha helical protein. There is no beta structure whatsoever. So that there is that to consider. Each alpha helical segment is labeled A through H. So if we start at the N-terminus down here, you'll see this first helix is helix A. You're going to see that we actually note in some cases bends. So here you have an alpha helix that bends. These are considered as actually two different alpha helices. The bend is what connects helix A with helix B, and that connector, that bend, is designated as AB. So if we keep on going here, we reach helix C, and then there's a linker. This is not a structured linker per se, but that linker connects helix C with helix D. So that linker is called linker CD, or just CD. So D then feeds into E, and you have another linker which would technically be designated DE. Keep on going through helix E to helix F, and you have a linker EF, and this system just keeps on going. Ultimately, you're going to reach helix H at the C terminus. So that is generally what how we're going to address or label the secondary structures within your myoglobin monomer. All right, so there are some unique ways in which we describe where an amino acid is in myoglobin, and the same is true for hemoglobin. This system that I'm about to describe to you is unique in that we don't really use it for most proteins today. We still use it for myoglobin and hemoglobin for historical reasons, but I suppose if you are the first to the field, you can invent the field. And that is what has happened here, which led to a less than systematic way to identify amino acids within myoglobin. So here, what has happened, the system that I am describing is one wherein an individual amino acid is designated by its position in a particular alpha helical segment. You'll see my description here says we can identify an individual amino acid by either its position in primary sequence or by its position in an alpha helix. The first of those two is the more conventional way to go about this, and just as an example, there's a histine residue in this F helix. Okay, so normally this histine, because it is the 93rd amino acid from the N-terminus, would just simply be called his 93. So if I started at the N-terminus, walked my way all the way through until I reached this histidine right here, this would be his 93. It is the 93rd amino acid from the N-terminus, and it is a histine, hence his 93. That sounds a lot like a radio station. That was not what I meant for it to sound like. So in the field of myoglobin or hemoglobin, the way that we refer to this his 93 residue is a little bit different. What they do is they look at what helix an amino acid is in. This particular histine is in the F helix, but not only that, it is the 8th residue in the F helix. So if I started at this end and I counted the number of residues from here all the way to the position of this histine, you would find 8 amino acids. So here, this histine is referred to as his F8. If I had something in the E helix, for example, like this residue right here, this is actually another histine, we would call that E, whatever residue it is from the N-terminal end of that helix. So here what I want you to see is that there are some unique myoglobin specific language that we have to pick up on. But more broadly, we are going to use the His93 sort of language because by convention, that is what we do for all other enzymes. Now, there is one other thing to point out here. I just used the term enzyme. Myoglobin and hemoglobin are not, in fact, enzymes. They do not catalyze a reaction. All they do is bind oxygen and deliver it from one place to another. So they bind and then dissociate. They're transport proteins, but they do not, in fact, catalyze a reaction. So for that reason, they are not considered enzymes. There is no catalytic function, no catalytic reaction that is being pushed forward. They're just oxygen binding and transport proteins. So please do not mix up Do not confuse myoglobin and hemoglobin for enzymes. They're not the same thing. Though when we talk about ligand binding, you will find that hemoglobin, myoglobin, they are paradigms for how ligand binding occurs and certain complexities that are related. But when we talk about catalysis, we will move to other examples. Myoglobin contains what we refer to as a prosthetic group. So if I move back one slide, you'll see this thing here, which I avoided any description of. Now we're going to talk about that. So that group is a heme group. And before I tell you anything more about the heme group is generally considered as a prosthetic group. A prosthetic group is something that binds to your enzyme or your protein and usually does so quite tightly, so the interaction is strong. There are certain cases where heme groups are actually covalently attached to a protein. In the example of cytochrome C, for example, the heme group there is covalently attached via a disulfide linkage to the protein. Here, that is not the case. This is still just a very strong interaction, but still non-covalent nonetheless. Now that I have said that, a prosthetic group, the goal of the prosthetic group is to provide functional groups that are not found in the standard 20 amino acids. So there is no real good contender for a functional group that can bind oxygen found amongst the standard 20 amino acids. So here we employ a small organic molecule, and by small I'm using that term loosely. This heme group here at its center has an iron that is chelated, and that iron is what binds oxygen. None of these standard 20 amino acids can bind oxygen in the way that iron can. So basically what your heme group is doing here is expanding the chemical diversity of the protein. That is broadly what prosthetic groups do. They allow for chemistry or molecular interactions that the standard 20 amino acids can't quite do. So, okay. Here, let's talk about the heme group for a moment. What I want you to see is that there is a lot of conjugation, so you'll see a lot of double bonds here. So the heme group absorbs light and does so quite strongly. That's where the color of your red blood cells come from. A lot of heme is there. At the center of the heme group, there are 4 nitrogen atoms. So these nitrogen atoms coordinate to a central iron. Now the iron is going to have 6 coordination bonds. 4 of them are going to be to nitrogen atoms that are part of your heme group, and the other 2 are above and below the plane of your heme group. Now, let me just use a little bit more specific language here. This is not just a heme. This is actually a porphyrin ring system. All of the conjugation in this porphyrin ring system leads to a planar heme group overall. So your iron is sitting right in the plane at the center of that planar porphyrin ring group, or the center of your heme, and then it can interact with constituents above and below the plane of that heme group. You'll see a little bit more about this in a moment. Let's take a look at the heme group in the context of myoglobin. Okay, so I'm going to throw a structural view up here for you, and if I back out, I want you to see I don't have the whole protein shown. If I did that, let me actually show you what that would look like. So here, this is myoglobin. For those that are curious, you can go to the protein data bank and search for these accession codes, 1A6N and 1A6M. You're going to find that one of these is going to be the deoxygenated myoglobin. The other is the oxygenated, so the oxygen-bound or the oxygen-free states. And if we look at the structure here, you'll see at the center is your heme group. We'll focus on that here in a moment, but the rest of this is just alpha helix. If you recall, per the labeling at the N-terminus, which you'll see I clicked on the end, the N-terminal valine 1 residue showed up, so that's how I know this is my N-terminus. That led into the first alpha helix, helix A. So you had a helix A, and then there was a bend that led into helix B, which is right here. Now I'm curious here, let's see what is in this bend. Well, this, as it turns out, there is a glycine right here, but this appears to be something of a natural bend. This is not a proline-induced bend per se. Just happens to be a bend that is likely due to stabilization or intramolecular interactions occurring elsewhere that induced a bend, but it's a bend. And so this bend here, remember we referred to this as the AB linker. And if you kept on going, you'd enter into helix C, helix D, then helix E here, and ultimately into helix F right here. Now this is important because we're going to focus on an element coming from helix F and then E on the bottom side. So at the center of hemoglobin is the heme group. If you look at the heme group here, you'll see it's flat, it's planar, roughly planar, and there is a histidine that comes from that F helix that is interacting with the iron that is at the center. So colored in orange, this is iron. I click on the entire thing. I was trying to click on the iron, but whatever. So at the center in orange is iron. In blue are nitrogen. So you'll see your 4 nitrogens here. And you'll see that this heme group is not covalently attached to the enzyme. However, it is covalently attached, and what I mean by this is not covalently attached at these outer corners of the porphyrin ring, but there is a covalent attachment between this histidine and that iron. That coordination bond is considered to be a covalent bond. Now, let me back out here and show you. Remember, so this histidine, if I go up to the primary sequence up top here, you'll see 91, 92, 93. Oh, this is the 93rd residue. This is hist 93. If I count from one end of the helix to the other, remember I already showed you this in a previous slide, this is histidine F8. So this is the 8th residue in this F helix. On the bottom side, you'll see this becomes important in a moment. There is another histidine, which is histidine 64. Okay, you'll see where that comes in a moment. And that moment is now. So on the top side, what is happening is the position of the iron is linked to the position of the alpha helix. So if one moves, the other moves. And you'll see why this matters momentarily. But on the bottom side, oxygen is going to bind. So oxygen binding to that iron is stabilized by an interaction with this nearby histidine on the bottom side. So there are 2 histidines. One is interacting directly with the iron. The other is interacting with the bound oxygen, which is shown here. This is O2. Okay. Now, just to give you a sense for what that looks like, you'll see that the position of the histidine on the bottom side interacting with the oxygen doesn't really change a whole lot. But what does happen, and it is very, very subtle, but you'll see that the histidine moves slightly down as when oxygen is bound. So just to play this back for you, notice in the deoxygenated state here, the iron is sitting slightly above the plane of the ring, but it moves down into the plane of the ring when oxygen is bound. So this is going to be very important, and you'll see why in a moment. The positioning of that iron above the plane or in the plane of the porphyrin ring, this is a very, very, very important distinction that is going to change the overall conformation of myoglobin and hemoglobin. So keep this in your back pocket for the moment. Okay, let's get back to the slide and view this a different way. So here, what I want you to see is this is just a schematic of what we were just talking about. And what you're going to see is that the porphyrin ring system is shown in pink here. So this iron sits in the plane of that porphyrin ring. His F8, or his 93, is sitting here above the plane interacting with the iron, and oxygen binds on the other side. Okay, so remember, the iron is interacting with 4 nitrogens, and the imidazole side chain of his F8, and when oxygen is bound, it is interacting with oxygen. Okay, so we have our oxygenated state and our deoxygenated state. Okay, so let's talk a little bit about this. So when the iron is sitting above the plane of the porphyrin ring, when oxygen is not bound, the coordination number for that oxygen is 5. Okay, so that doesn't really mean a whole lot to you just yet. But what's going to happen is when oxygen binds, it's going to change the coordination geometry of that iron. It's going to move down into the plane of the ring, as I've already shown you. And this is going to induce a change in the electronic properties of the heme as a whole. And this is actually going to account for the change in color from that dark purple for oxygen depleted blood to the bright red of oxygen rich arterial blood. So this is something that is incredibly important. You have seen this. You can see this right now if you look at your arm. So you will note that the electronic properties of that heme group are centered on the iron and the coordination geometry of the iron. And this is going to change the behavior and the structure of that heme group. So some other things to take note of here. In a previous lecture, I told you that the heme group is largely solvent inaccessible. And that is important because if this iron is to become, if it undergoes oxidation to take it from the iron 2 state, which is what is shown here, to the iron 3 state, iron 3 cannot bind oxygen. So the heme needs to be solvent inaccessible to prevent the occurrence of iron oxidation, which would also prevent binding of oxygen. This is really very significant. Remember, if you go to the grocery store and you find brown colored meat, this is just simply due to the oxidation of that iron. Now, there's some other things to account for before we move forward, and that is really that oxygen is not the only ligand to which myoglobin can bind. Other small molecules like carbon monoxide, nitric oxide, hydrogen sulfide, these can all bind to heme groups just as you are seeing right here, in the same way that oxygen is binding to the heme group. However, the difference is that these other compounds actually can bind with much, much higher affinity than oxygen. And this is really significant because what that means is that something like carbon monoxide can bind to your heme group with a higher affinity, therefore it can outcompete oxygen for binding to that heme group, which is then going to lead to toxicity. I think we can all appreciate that carbon monoxide poisoning is bad, and it's not in fact poisoning. What it is more of suffocation, because carbon monoxide, it binds to your heme group, prevents oxygen from binding, therefore you are starved for oxygen under those conditions. In fact, carbon monoxide has a 200-fold greater affinity for hemoglobin binding than does oxygen. So that is something that is very, very significant here. We have to understand that oxygen is not the only thing that myoglobin and hemoglobin can interact with. There are other molecules, and it is very important to recognize that fact. For fear of sounding like a broken record, let me go back into the structure of myoglobin and make a little bit more fuss about the difference between oxygen bound versus oxygen free myoglobin. What I want you to see here is that there is a very subtle global change in structure here that shows up when oxygen binds versus when it is not. So here, this is the deoxygenated myoglobin. You should already be able to look at this and see there's no oxygen there, and that iron is sitting just above the plane. You can see it peeking its head up there in orange. So, okay, there it is. Now, if you look at the oxygen-bound structure, let me just sort of flop back and forth here. What I want you to see is that there are some subtle structural changes. In particular, if you would focus on this helix here, this F helix that I am circling with my cursor, you'll see that there's a slight downward movement when oxygen is bound. So let me just replay that for you. Here's apo, no oxygen bound. Here's with oxygen bound, and that helix gets pulled down a little bit. There's a slight downward movement. See? Okay. Now, how does that happen? Why does that happen? Well, it all centers on his F8. So when that iron gets pulled down into the plane of that porphyrin ring, when oxygen binds, the histine gets pulled down with it. So here, you should see, when that iron moves down into the plane, oxygen binds, there's a change in the electronic environment of the heme. Now the iron is in the plane of the ring, the histine moves down. That drags this helix down with it. There are some other structural changes that occur too, but this is going to be important, and you'll see why I'm making a big deal about this a little bit later. Now, some other things I want to highlight. So here, oxygen is our ligand. Oxygen is what is bound. If I superimpose these, you should see that oxygen binding versus the free state, there is a slight conformational difference, but oxygen is still bound in this case. So let's compare this to some other things. Here's oxygen. Now let's look at carbon monoxide. So this is bound carbon monoxide, and you should take note of the fact that it looks a lot like your oxygen bound state. You'll see here's oxygen, here's carbon monoxide. There are some very subtle differences, yes, but in both cases, the iron is in the plane of the heme, so it is, for all intents and purposes, binding. Carbon monoxide is binding much like oxygen does, except it does so with a much, much greater affinity. There's also another contender here. This is cyanide. So here, this is cyanide, and you'll see, again, the iron is in the plane of the heme. Cyanide binds also to myoglobin and hemoglobin. So you have many, many options in that are, you have small molecules that are potential poisons and function through displacement or out competing of oxygen for binding to myoglobin or hemoglobin. So you've got carbon monoxide, cyanide, hydrogen sulfide, various small molecules like that. And examination of structures show that binding is very similar to oxygen, but this is a problem because they bind with higher affinity, therefore prevent oxygen from binding to these oxygen transport proteins, which we so desperately need to perform respiration. Okay, so myoglobin, hemoglobin, they are incredibly important for mammals such as us, but they are not the only players in town. They are not the only proteins that can bind oxygen. Other organisms have evolved different methods to do so. So one example here is shown to you. This is the general family of hemocyanins. So here what you have is basically a copper-centered mechanism. And you'll see in the binding site there are histine residues that really it's a symmetric binding site. You have 3 histine residues on one side, 3 on the other. 2 coppers overall become coordinated, and oxygen binds across those coppers. So this is important because this is not your heme-based binding mechanism like you saw for myoglobin or hemoglobin. And this represents an alternative to heme-driven oxygen binding. So the result here is actually that the blood of organisms that have hemocyanins is blue. So you'll see the common example are horseshoe crabs, which are what I'm showing you here. Horseshoe crabs have blue blood, so perhaps they should be the mascot for MTSU because they literally bleed blue. I make that joke every semester and I crack myself up every semester. But horseshoe crabs bleed blue. They have blue blood due to the presence of hemocyanins that transport oxygen. In fact, what you're looking at here is horseshoe crab blood. They are actually being studied for potential clinical applications. Now, in all organisms, it is really important to understand, in all organisms that require oxygen, you're going to have some sort of transport system in place. Oxygen is not terribly soluble in water. It is, if you think about it, it's a nonpolar molecule. The dipoles that would normally make it polar are symmetric and cancel each other out. So oxygen, O2, is not terribly soluble in water. And for that reason, the way that we transport gases like oxygen is by binding to a soluble molecule that can transport it. So in the case of hemoglobin, myoglobin, hemocyanins, oxygen is going to bind to your protein, which is soluble, and it is then going to be transported through the blood to wherever it needs to go. So if you are an organism that does any amount of oxidative metabolism or respiration, you are going to have an oxygen transport protein that is going to facilitate movement of that oxygen from one place to another. So, moral to the story, the takeaway point here, hemoglobin, myoglobin, they are not the only way to move oxygen between two locations in an organism. There are other proteins that can do this also. Usually, this is going to occur with a metal-assisted process, but just know that the oxygen is not going to probably move by itself. Up to this point, we have only talked about how oxygen binds to the binding site for myoglobin. We have focused on the heme group without a whole lot of consideration of what else is going on there. So now we're going to pause for a moment and actually look at an enzyme as an example here to really get the point across that binding of a ligand at a binding site or an active site in the case of an enzyme, it is going to involve interaction with specific amino acids, but the shape and overall size of that active site have to be complementary to the ligand that is trying to bind to the site as a whole. So basically what I'm trying to tell you is that if your active site or your binding site is not of complementary shape and size, it is not going to accommodate a ligand being stably bound at that site. So as an example, let's look at chalcone synthase. I have a typo here apparently. This should just be chalcone synthase. But here, the binding site for this enzyme has been synthetically, or rather genetically, manipulated. So here I'm showing you a view of the enzyme bound to a substrate, naringenin. So here, this is a very unique binding site because it's actually somewhat, it's almost like there are two lobes that lie perpendicular to one another. There, you'll see the substrate here sort of extending backward. That would be one lobe, and then there's one that lies perpendicular that contains some of the catalytic elements of the active site. So this is a polymerization reaction where the very earliest substrate that's going to bind to this enzyme is going to basically occupy about half the space, half the volume of this, this lobe of the active site, and then it's going to grow outward. So this is interesting because what you can do at this glycine 256 position is start making mutations that incrementally increase the size of the R group at this position. By doing this, you are decreasing the volume of this particular lobe of the active site, and you prevent growth. You prevent, basically growth of the product that is going to be formed. The product is going to occupy more volume, more space than the initial substrate. And so if you increase the size of your side chain at this 256 position, you then restrict how big your product can be. So what's been done is this glycine has been mutated to an alanine, valine, leucine, phenylalanine with results that I'll tell you about on the next slide. If you look at the surface representation of the binding site here with each of these mutations made, what you're going to see is that 256 position, when an alanine is there, it occupies more volume. You can see the volume highlighted in blue here. It occupies more volume than a glycine would, sure. But when you convert that to a leucine, you occupy even more volume. A valine, a little bit less than a leucine. So we're basically going from alanine, valine, leucine, then finally to phenylalanine, where you are, with each next mutation, increasing the volume that side chain occupies, until ultimately, with phenylalanine, you're basically hitting almost the catalytic cysteine residue at position 164. And so with each mutation, you are changing the active site volume. We can also calculate the active site cavity volume. For the wild type enzyme, we see that the active site cavity is 605 cubic angstroms. Now, before I proceed, let me reiterate here that this portion of the active site cavity accommodates a lengthening product that is being formed. This is a polymerization reaction where the product is growing in size relative to the reactant. So by decreasing the volume of this cavity, we are restricting the size of products that we can make. So this is going to be really important, and I'll actually show you the mechanism here in the next slide. But here, the wild type enzyme has an active site cavity volume of 605 cubic angstroms. If we take that glycine to an alanine, that reduces ever so slightly to 601 cubic angstroms. If we introduce a valine, we drop further to 590 cubic angstroms, a glycine, 572, and then ultimately that phenylalanine drops at the furthest to 352 cubic angstroms. So here we are incrementally increasing the side chain size and thereby causing a decrease in the active site cavity volume. And this is very important because this is going to severely restrict the elongation or cyclization pocket of the active site cavity, and it places restrictions on how big your polymerized product can become. Okay, let's actually look at the mechanism, tie it to the actual data. So first, if we look at the mechanism for chalcone synthase, what you're going to see is that you're going to start with malonyl-CoA, and then there's going to be a condensation reaction with this molecule that is going to lead to the formation of a triketide molecule. And what can happen is this triketide can undergo cyclization to basically proceed down two possible paths. There's going to be a non-productive path, which is going to give you this molecule, the molecule that is shown here to the right-hand side, or you can proceed down another condensation path where this triketide undergoes condensation with another malonyl-CoA to give you a tetraketide, shown here. Yet again, the tetraketide can go down a non-productive path or proceed all the way to form chalcone, which then undergoes spontaneous rearrangement to naringenin here. So this is the final product. So the going from reactant to product involved a series of stepwise condensation reactions that led to increasing polyketide lengths. So if our active site, where all of the action is happening, is not big enough to accommodate that growing polyketide, we can't actually form naringenin. If we actually look at the data here, so this is thin layer chromatography, you can see for the wild type enzyme, here marked as CHS, the wild type enzyme forms a mixture of naringenin, so 62% naringenin, but still about 33% of this C-tau off pathway product. So even the wild type enzyme isn't perfect. That's fine, but that gives you your point of reference here. Now, when you introduce that alanine, remember the alanine here at position 256 decreases the cavity volume ever so slightly, but you'll see a decrease in naringenin formation going from 62% to 42%. So here we see a drop in naringenin amount. Most of it has gone to form that transition, the difference, most of it has gone to form more C-tau. So the alanine made it so that you couldn't form the final product ever so, it's not quite the same shape. The active site is not shaped exactly the same way. So your final product distribution relative to C-tau is altered ever so slightly. If you introduce a valine, similar story. But once you make that next jump to leucine, there, if you remember, if you look back at the active site cavity volumes, that was a pretty big jump. So your active site cavity volume dropped pretty significantly going from valine to leucine. So there, we don't form naringenin anymore. We also don't form C-tau. What we do form is this molecule. Yeah, okay, we form this molecule as well as methylpyrrone. So basically, this is the result of not being able to take the step from triketide to tetraketide. You have restricted the active site cavity volume, so there's not space to accommodate that next condensation step. So you'll see for leucine and phenylalanine at the 256 position, we form a mixture of species of triketide related species as opposed to the tetraketide related species. So my point in telling you this, when we think about the binding site for any protein, prosthetic groups most certainly play a role. They provide functional groups that are necessary to do chemistries that the enzyme, these standard 20 amino acids may not be able to do on their own. But the shape, the size of the binding site has to be able to accommodate the reactant as well as the product and do so in a way that stabilizes all of the different reactants so that you will get productive product formation. You'll see in Chapter 6 how this really occurs, but at this stage I'm trying to introduce you to the idea that shape and size are very important drivers of that initial protein-ligand interaction. Now we're going to pivot to looking at the thermodynamics that describe ligand binding to a protein. So here, we're going to walk through a little bit of math, but the point here is to get you to understand how to interpret some of the properties that are commonly used to describe the strength of interaction between a protein and a ligand. So what we're going to assume here first is that binding of ligand to your protein is reversible and can be described by a reversible equilibrium. So that's going to be very important because that then allows us to calculate an equilibrium constant, which can then be used to calculate some other terms here that I'm going to show you now. We're about to pivot to pen and paper. I'm going to derive this for you. I've got this shown here, but I'm going to just go ahead and derive this so that you can sort of walk through and see where all of the assumptions go into this. Okay, let's go ahead and move over. And all right. So now, yes, I have a model protein here that I was using as a paperweight. Let's write out what it is we're talking about. So my protein binds one molecule of my ligand and does so reversibly to form the PL complex, the protein ligand complex. Okay, so now we can write an equilibrium constant. So this can be described by an association equilibrium constant, just like any other reversible reaction can. So Ka equals the concentration of PL over concentration of P times L. So here, notice my stoichiometries are all 1-to-1. So one protein, one ligand, gives me PL where one ligand is bound per protein. This is very similar to the case of myoglobin. So here, this is my equilibrium constant. And now what I'm going to do is use this information to derive a parameter that is referred to as the fractional saturation. Now, before I do this, I want to remind you of something. This equilibrium constant, remember, all this is an equilibrium constant. An association equilibrium constant that's what this is showing you. If I take the inverse of this, so that reactant concentrations are on top of product concentrations, so the concentration, if I were to do, say, 1 over Ka to give this, all this is my Kd, my dissociation constant. So all this, the association equilibrium constant is related to the dissociation equilibrium constant simply in that one is the inverse of the other. That's all it is. But whether you're using Ka or Kd, all we are looking at is the quantitative expression of the equilibrium between the complexed, and what I mean by that is the bound state and the unbound state. So all this ever is a ratio of one thing to another. That's all this is. So don't try to overcomplicate this. It tells us how much bound complex we have versus free ligand. This is important because what this is going to tell us is really the strength of the interaction. If more product complex or more PL complex is formed here, that tells us probably that the interaction is stronger than, say, a case where you have more free ligand than you do product, or rather protein ligand complex. Okay, so now fractional saturation. What is this? What exactly am I looking at? So keep this equation in mind. Know that I'm going to come back to the Ka. Okay, so my fractional saturation, written as theta, or sometimes you'll see it written as x bar, the mean, or rather the extent of binding. It depends on what who you're talking to. Fractional saturation, extent of binding, they're fundamentally defined in the same way. It's the concentration of binding sites. Let me just put that concentration bracket there. Binding sites occupied over the concentration of total binding sites. Okay, so to say that again, the fractional saturation, the extent of binding, all it is the concentration of binding sites that are occupied over the concentration of total binding sites. So what that should tell you is that this term can only scale from 0 to 1. So the concentration of total binding sites, if all binding sites are occupied, that means that this term is going to equal that and give you a ratio of 1. If no binding sites are occupied with the ligand, then that is a term of 0 over whatever value this is, which is overall 0. So you're only going to scale from 0 to 1. Don't forget that. But also, once we start talking about math, just remember that it's always qualitatively or conceptually viewed in this way. All the stuff on top is going to be, basically all of that equals the concentration of occupied binding sites, and all the stuff on the bottom just equals the concentration of your total binding site. That's all that you're looking at here. Now, if we go back to this scheme, I have everything I need right here. So the concentration of occupied binding sites is given by the concentration of PL, and the concentration of my total binding sites is given by the sum of P plus PL. Basically, the protein here contains all of your binding sites, so the concentration of total sites is contained as the basically sum of the protein in all states. So this can then be written as concentration of PL over PL plus P. So that's all that your fractional saturation is. Now, we're not going to leave it here because this is not great for actual use. It can be difficult to determine all of these parameters simultaneously. So what we're going to do is take this further into a more usable form. Okay, so if you remember, what we want to do here is derive an expression that we can use experimentally that has the fewest parameters possible. So the easiest way to do that is to convert this expression into an expression that has really two parameters, the equilibrium constant and the free ligand concentration. Most of these experiments are going to be done, the actual binding experiments that we'll talk about, are going to be done under conditions where your ligand concentration is so large that you can assume your free ligand concentration roughly equals your total ligand concentration. So that's really going to be one of the assumptions that goes into this. Okay, so what am I going to do with this? What do I do? Okay, so theta, fractional saturation, is going to now equal this expression just substituted. So if I remind you, my Ka term equals the concentration of PL over P times L. So what I can do is solve for PL. So then concentration of PL equals Ka times P times L. I didn't really give myself enough space there. But now theta equals the concentration of, let's see, write this out as Ka times P times L over Ka P L plus P. Okay. So now I have this thing. This is great, but this is not the most simplified view that I could arrive at. I still have protein concentration terms that cancel out, in fact. So this will cancel, that'll cancel, this will go to 1. So we can rewrite this as Ka times L over Ka times L plus 1. Okay, so this is really your most simplified view here. And you'll see what this looks like in a moment. But let's just say that this is not where you want to stop. Let's say you want to express this in terms of your dissociation equilibrium constant. So what you can then do, so I'm going to rewrite the expression in terms of Ka, K times L plus 1. Okay, so what you can do, remember, your Ka is related to your Kd by the inverse. So if I multiply this by 1 over Ka over 1 over Ka, okay, so what I'm going to get here is something that looks like this. Concentration of L over L plus 1 over Ka, which equals L over L plus Kd. So you'll sometimes see it like this. If you spend enough time with me, you'll see that I usually express it like that, but they are interchangeable. I just wrote the reversible equilibrium signs there. they are reversible. So you can express the fractional saturation using either your association or your dissociation equilibrium constants. Now, one thing to take note of here is that, as you will see in a moment, both of these functions are going to give you, if you actually look at the data, you are going to see a rectangular hyperbola. That's, I will be using this term a lot, but that is going to be defined mathematically by this function. Now, another thing to take note of here, if you convert between these two states, these two ways to express the fractional saturation, the equilibrium constant is going to have different units. Your Ka has units of 1 over molar, so 1 per molar. Your Kd is going to have units of just molarity, so don't mix up your units here. All right, so now let's move back and continue on. Now if I plot that function that I just derived, fractional saturation, what you're going to see is a function that looks like this. So here I am plotting fractional saturation on the y-axis and ligand concentration on the x-axis. And you'll see an asymptotic approach to a value of 1. So as I increase the ligand concentration via a function that is a rectangular hyperbola, I'm going to asymptotically approach 100% fractional saturation. So if I increase the concentration of ligand high enough, I will at some point fully saturate my enzyme. Now, this plot is very useful because what it tells you, what it allows you to qualitatively deduce, is the actual value for your binding constant, your equilibrium constant, describing that protein-ligand interaction. So when you are at 50% fractional saturation, if you extrapolate back to the x-axis, the direct extrapolation gives you an estimate of your dissociation equilibrium constant, Kd. If you take the inverse of that, you can get Ka. 1 over Ka equals Kd. So here, what I want you to understand is that the Kd determined in this way is equivalent to the molar concentration of ligand at which you have half of your protein binding sites occupied. So again, when your ligand concentration equals Kd, 50% of your protein, your binding site, is occupied by ligand. As your ligand concentration falls below your Kd, less of your protein has ligand bound to it. And in fact, in order for 90% of your available ligand binding sites to be occupied, your ligand concentration must be 9 times greater than your Kd per the predictions made by the rectangular hyperbola. Now, let me point you to this plot right here. This is more of what you should expect to see when we talk about oxygen binding proteins. So here, the x-axis, which would normally just be ligand concentration, is replaced by the partial pressure of oxygen. So this is an important thing to note because the concentration of dissolved oxygen is notoriously difficult to determine. It's really hard to pin that number down because of the poor solubility of oxygen. So what has been historically done is that the concentration of oxygen is assumed to be proportional to the partial pressure of oxygen under a certain set of conditions. So here, what you're going to see is that your PO2 is a stand-in for your actual ligand concentration. This doesn't mean that the concepts that we'll talk about are invalid across the board. I just want you to know that this is a different way to express the concentration of a gas. That is all. Now, let's add some complexity here. So let's say that ligand doesn't bind just at one site, but there's actually now a possibility for binding at multiple sites. So there are multiple ligand binding sites per protein or per protein complex. Now we have the possibility for something called cooperativity. We have the possibility for cooperative binding. So before I go into great depth, what I want to just go ahead and tell you is that cooperativity, this is a quantitative way to assess the influence of ligand binding at a first site on secondary binding events. This can be a positive influence or a negative influence, but all cooperativity is a quantitative way to really describe how binding a first ligand binding event, so you bind that first ligand to your protein, how that event influences secondary events. So you will see cases where binding of your first ligand actually makes secondary binding more favorable. So you bind that second ligand more favorable than the first. You can even see cases where binding of your first ligand makes the second ligand bind more favorably, which makes the third ligand bind more favorably, which makes the fourth ligand bind more favorably, and so on and so forth. And usually when we talk about cooperativity, this usually is positive cooperativity, but it can be negative cooperativity. You can see that binding of that first ligand discourages binding of the next ligand, which discourages binding of subsequent ligands. Usually, this is going to be structurally related. You're going to see that when your first ligand binds, perhaps there's a conformational change in your overall protein complex that makes it harder, sort of distorts those other binding sites, or perhaps makes those binding sites become restructured in a way that is more supportive of ligand binding. So cooperativity is something that we really need to understand, and the way that we quantitatively do this is usually tied to something called the Hill coefficient. So here written as N, the Hill coefficient. This is a quantitative measure of cooperativity. It's actually referred to as an interaction parameter these days. So what do I do with this? Okay, so let's first take a look at the math behind the expression I'm showing you here. I'm going to move back over to my pen and paper format. Okay, let's take a look again at this binding equilibrium. This is what I just showed you on the previous slide, our reversible binding of a ligand by protein. But now, let's change the stoichiometry. So let's say our protein binds N ligands to give a PLN complex. So per the derivation I'm about to show you, we are not going to allow for stepwise events. This is basically going to be protein binds all of our ligands and goes to the fully saturated complex, and we're going to use this as a measure of cooperativity here. So we're going to turn back to our fractional saturation route that we just went through after I write out my equilibrium constant. So you see Ka now equals concentration of PLN over concentration of P, but now my ligand concentration term is raised to the nth power. Okay, so let's do this again. Let's write out fractional saturation. So theta equals PL over concentration of PL plus concentration of protein. So again, this is the concentration of binding sites that are occupied over the total concentration of binding sites. And let's go ahead and plug in our terms again. So PL is going to equal, or rather PLN is going to equal Ka times P times L raised to the nth power over Ka times P L raised to the nth power plus the concentration of P. Let's cancel our P terms out. This becomes 1. So now this is Ka times L to the nth over Ka times L to the nth plus 1. Okay, so if you want to put this in the form of the Kd instead of Ka, I'm just going to skip some steps and write this. Remember, this is just basically multiplying this times 1 over Ka to convert this into Kd units. And then theta becomes L to the nth over L to the nth plus Kd. Okay, so this is exactly what is shown to you on this slide. Okay, so now what you should see is that the Hill coefficient N, all this is a measure of how many ligands are bound to our protein. It is effectively the stoichiometry, mathematically the stoichiometry of ligands that are bound in the fully saturated protein complex. Now, I will go ahead and point out that it has been demonstrated across many, many different examples that even though the Hill coefficient mathematically should estimate the number of binding sites that ligand is binding to, in fact, that is not always correct. the more correct interpretation, and in fact the more conservative interpretation, is simply whether or not positive cooperativity occurs versus negative cooperativity. So if you see a Hill coefficient that is greater than 1, that tells you that there is positive cooperativity. And if you see a Hill coefficient equal to 1, you can either conclude that you have negative or non-cooperativity taking place. Now let's look at the actual data that you might expect to sort of get a feel for further interpretations. So if you look at fractional saturation as a function of oxygen concentration, expressed here in terms of the partial pressure of oxygen, you'll see that when the Hill coefficient equals 1, this should be non-cooperative behavior. There's no cooperativity in ligand binding. You will see that we have this rectangular hyperbola that you would fully expect. Based on the derivations I just showed you, a Hill coefficient of 1 should tell you that there is no cooperativity, one ligand is binding, theoretically, and that gives you hyperbolic data. Now, I want to give you a caveat. It is possible that you have a protein complex or a protein with multiple binding sites, but each binding site functions independently and binds ligand with the same overall dissociation equilibrium constant, or affinity, as I will call it sometimes. So there, you would have no quantitative measure of cooperativity. You wouldn't even know that each protein had a stoichiometry of greater than 1, and your Hill coefficient there would reflect non-cooperativity and wouldn't really tell you anything about stoichiometry. So the reason that the Hill coefficient is more conservatively used to infer cooperativity is exactly for the reason I just laid out. You can have binding of multiple ligands to one protein, but just with independent binding that is not influenced by other binding events. They all would bind with the same affinity, and you wouldn't know any difference unless you determined directly the stoichiometry of binding. Now, let's say you do have cooperativity. So your Hill coefficient here, n, is greater than 1. What you're going to see are sigmoidal data, so S-shaped binding curves. So here, when n is 2.8, you see an S-shaped binding curve where at low concentrations there's not a whole lot of ligand bound, but then you see a steep increase, a sigmoidal increase in the amount of ligand bound until you approach full saturation. The Hill coefficient itself determines the slope of that transition. So if the Hill coefficient is 4, you'll see a steeper transition relative to when the Hill coefficient is equal to 2.8. So again, the Hill coefficient is a measure of cooperativity. It tells you to some extent how cooperative ligand binding is. So I've been talking a lot about this Hill coefficient thing, but I haven't really told you where the name came from. And in many of my courses, I like to humanize the science where I can. So I want to give you a sense for who Hill is in the Hill equation, which is what I just derived for you. So Archibald Vivian Hill, he is Hill of the Hill coefficient. Archibald Hill is widely considered to be one of the founders of biophysics. And this is not meant to be scary. This is meant to just really remind you that biochemistry is highly interdisciplinary. So Archibald Hill, he won the Nobel Prize in Physiology or Medicine in 1922 for his work in the field of physiology. Now, this is really interesting because his entry into physiology is going to be somewhat unorthodox, and I'll tell you about that momentarily. So his first publication was in 1909. He derived various different types of binding equations, and his first paper was really centered on what would be known later as the Langmuir equation, and that was his very first paper. He published the Hill equation in 1910, and there he was looking at the study of ligand binding by hemoglobin. He was looking at basically oxygen binding to hemoglobin, and he needed to derive a way to quantitatively describe it, and so he did. And just to make the story complete, he liked to enjoy, or he enjoyed playing hockey and going on long hikes as a child. This will actually be related, and you'll see why in a moment. But Hill, he entered into physiology via a very unorthodox path. So at the time, entry into any given discipline required extensive study in that discipline. So if you wanted to go into chemistry, you needed a background in chemistry. If you wanted to go into physiology, you needed a background in physiology. Hill's training, he had training in chemistry, physics, and math, but not at all in medicine or physiology. So he had no training in biology or anything like that. That would be basically like me saying, oh, I'm going to do clinical studies right now as a biochemist with no formal training as a biologist or as a clinician. That's just something that is very unorthodox, but it is very cool at the same time because Hill brought a unique interdisciplinary perspective to a field that was largely homogenous at the time. So imagine, the field of physiology at the time was dominated by people with training in physiology, but they did not necessarily have that same training in chemistry, physics, or math. And so he brought a very unique perspective to the field, which is a very good example of the interdisciplinary aspect of biophysics and enzymology as a whole that you'll see develop with time. So I came across this description of Hill and his working conditions at some point, and I like to share it with students now because I think that it really well summarizes him and his motivations, which I think can be very inspiring to students at your stage. So first, this description was written by a German researcher that was visiting the lab in which Hill was working. So the German style was very different than the English style. So first, I'll just highlight a few points here. The English method of working is greatly different than the German. So the laboratories themselves, the physical location where the work was being done, the laboratories themselves were housed in older buildings. They were rather homely and modest. So this is, you can still see this in some places, and there are locations in the United States that are modeled in the same way. The laboratories looked like houses. They were actually housed in former homes. So you looked at a house and you looked at a laboratory and you didn't necessarily know from the exterior if it was someone's home or if it was a lab. The German labs were very different. You knew what you were looking at. Now, the culture of the English was also very different. So if I hop down to the bottom asterisk here, you'll see they all came into work just when they felt like it. Only when they were to teach students did they arrive punctually. So this is true in many labs in the United States now and elsewhere, that graduate students, researchers, postdoctoral fellows, generally, unless there's a lab culture that states otherwise, people come to work and then they go home. And perhaps they work long hours, sure, fine, but it is not uncommon to see people show up at noon. They come to work when they come to work, but when they're at work, they do the work. And I know that I'm using the term work a lot here, but the point here is that there's not a set time in which people had to show up to work. They came, and they came when they felt like it. And if you prefer to sleep in, you sleep in. If you want a late breakfast, that's fine. You come to work when you get there. But once you're there, you're going to work until you're done. And then you're going to go home and do the whole thing again. This is very different than the 9 to 5 sort of schedule that the Germans were used to. They came to work at a set time and they worked until a set end time or until they were done. And you had to be at work with punctuality. And so here, that last sentence, only when they were to teach students did they arrive punctually. I think we can all appreciate that you have to come to class on time. And that applies to the teacher as well. So if you have to teach, fine, you have to come to work at a set time. But if you're coming just for research, no, not so much. So there's a much more informal atmosphere to the research itself in the English style relative to the German. Now, that was good because that informality allowed, in some cases, for students to enter into fields that maybe they didn't have the full academic preparation for. So Hill is that, a very good example of that, because remember, he was trained in chemistry and physics, but not in physiology. So that allowed him the possibility to enter into the working in a physiology lab. But he is actually a very cool example because, to draw you down here, you'll see the description. He was a great enthusiast for various sports, particularly running. Once while he was running, the thought occurred to him, how does the muscle machine work? And this led him to the physiology laboratory where he has worked ever since. So a question of basic curiosity, how do my muscles work? How does the muscle machine work? That basic curiosity led Archibald Hill into the field of physiology from his training in chemistry, physics, and math, and he applied those skills to just this very basic question. And that led to the development of the Hill function, ultimately the awarding of the Nobel Prize in Physiology. So my point in telling you all this is I want you just to really understand that it is okay to do science because you are just curious, because you just want to understand how things work. That is the basic tenet by which some of the most important science has been done. So don't ever let that drive to just understand how things work, that basic curiosity, don't lose that, because once you've lost that, the spirit of science is also lost. And I think Hill here is a very good example of just doing the science that interests you. Okay, so now let's take a moment to talk about the actual Hill model. I have been tiptoeing around this, but let's actually talk about the Hill model that was derived by Archibald Hill. So in the Hill model, I have shown you this written out, but here we're going to step by step sort of walk through some of the aspects here. So in the Hill model, a macromolecule with N fully saturable ligand binding sites is treated as though there are only two states. There's your free macromolecule, your uncomplexed macromolecule. Here I should note that I'm using the term macromolecule in place of protein because the Hill model is also applicable to other types of ligand, other types of molecular interactions. So think DNA interacting with a ligand, or even synthetic molecules like heme. They can be treated as macromolecules. So the macromolecule here is the larger molecule that is capable of interacting with multiple ligands. Okay, so here we have two states, our uncomplexed macromolecular state M, and the fully ligand-bound state, the saturated state, MLN. Okay, so first, I should note that this is not physically realistic. This is, at the time, the assumption that made the analysis accessible to Hill and is still used to this day, but in reality, you're probably not going to go from completely free to fully bound without stepwise intermediate steps. So just know that there are some physically unrealistic assumptions that go into the Hill model, but it still provides a decent approximation of cooperativity. So if we write out the equilibrium reaction, you see something like I have already shown you. M plus NL goes to MLN. So we have macromolecule, N number of ligand molecules, and macromolecule bound to N number of ligand molecules. You can write out your equilibrium constant as I have already shown you. And this is written as a KD, not as a KA. So just know that. And we can, through some mathematical magic of sorts, generate a linear function. So here what we have is an expression of moles of bound ligand per total sites as a saturation fraction. And so this is technically what we call the Hill equation. Now, I have already shown you another form of the Hill equation, but this is just a more complicated linearized form of the Hill equation. If we actually look at the Hill equation in action, you'll see you have y equals mx plus b. This is a y equals mx plus b function. So if we plot y as a function of x, which is log ligand in this case, you'll see that the slope of your linear region here is going to be proportional to n. So the linear region of your Hill plot has a slope of nh, or your Hill coefficient, and this is an approximation of your cooperativity as I have talked about. So just to give you a little bit more information, under some circumstances, you can actually tie your Hill coefficient to the number of ligand binding sites. Okay, so the limitation here is that your Hill coefficient is always going to be less than your number of binding sites. You can approximate the number of binding sites that ligand is occupying, but it's not going to tell you how many sites you actually have. But that is only applicable under certain conditions. So just know that the more conservative interpretation of your Hill coefficient is as a measure of cooperativity. So just please know that I'm trying to give you the more conservative interpretation of the Hill coefficient. So in this class, we are going to use it as a way to measure whether or not there is cooperativity, and we're not really going to focus on the sub cases in which it might allow for estimation of stoichiometry. So just please, please, please remember that. Okay, now if we look at some experimentally determined protein dissociation constants, so these would be KDs, what you're going to see is that KDs for biological systems are going to vary across orders of magnitude. You're going to see enzymes like avidin binding of a ligand biotin with affinities that are in the order of 10 to the minus 15th molar. So that is incredibly strong in terms of its interaction strength. That is tight binding, as we call it. On the other end, you'll see calmodulin interacting with calcium with a KD on the order of 10 to the minus 6th. So that's a lot weaker. That's micromolar binding affinity as opposed to the picomolar and femtomolar binding affinities you see on the other end. But you're going to see binding affinities that span all across this range. And usually this is going to be tied to the function of the protein-ligand interaction. Why are these things interacting? You'll see in some cases, like in the case of biotin avidin, that there is a specific physiologic purpose to binding with such a high affinity. Other things like think protein binding to DNA through, like repressor, transcriptional repression, things like that. You want to bind tightly, but you also want to be able to dissociate at some point. And so you may have intermediate binding affinities. But in the case of enzymes interacting with substrate, you inherently want a lower binding affinity there, because if you bind too tightly, you can't ever then convert substrate to product and actually let go and do the whole thing again. So you need lower affinities there. But here you should get a sense for the span that affinities range. So you go from 10 to the minus 15th down to 10 to the minus 5th, sometimes 10 to the minus 3rd millimolar KDs, but those are very, very low binding affinities. So just know that you span orders of magnitude here. Now I want to provide some introduction to the use of these graphical representations that we've already talked about. I have introduced you to what the typical plot would look like, where you have fractional saturation on the y-axis and concentration of ligand on the x-axis, where in the case of hemoglobin or myoglobin, you're looking at percent O2 saturation as a function of the partial pressure of oxygen. So here, there's a little bit more that can be said. First, if you looked at the midpoint, the 50% saturation point, you're going to see, if you find 50%, extrapolate over to this plot. This is actually for myoglobin. and then extrapolate back to the x-axis, you will find what is called a P50 value. And so when we are using the partial pressure of any gas, this is not going to be expressed in the KD is not going to be expressed in terms of molarity. So for that reason, this is the partial pressure at 50% saturation, the P50 value. So this for myoglobin is 2.8 torr, where here your pressure is being expressed in units of torr. And for hemoglobin, if you extrapolate over, you'll see something that is greater than 20 torr. It's just shy, I guess, about 25 torr. Now, one thing that I want to start to familiarize you with here is how to read beyond just looking for the KD. What I want you to be able to do here is, when it is annotated, make predictions about physiologic conditions. Okay, so just as an example, in arteries where you have a lot of oxygen, so here the arterial PO2 is quite high. If you find that concentration, extrapolate up to the binding curves here for myoglobin and hemoglobin, you'll see that they are both near saturation. So the here purple, if I extrapolate over to the y-axis, I'm at about 90% saturation. Myoglobin is going to be fully saturated. Cool. But if I take either of these proteins down to working muscle, in working, vigorously contracting muscle, I'm using O2 to facilitate aerobic respiration, so my oxygen concentration is lower than in those arterial conditions. And you'll see that under those conditions, where I guess we're at about, say, 10 or 15 torr-ish, myoglobin is going to be less than 80% saturated, and hemoglobin is going to be about 10% or less saturated. So the degree of saturation, the fractional saturation here, depends on the concentration of ligand that's available. To build that example, if we went in the same tissue, going working to resting in muscle, you'll see the concentration of oxygen is much higher in resting muscle than working muscle, which then means that my myoglobin is about 90% saturated, hemoglobin is about just over 80%, but the degree of saturation here, going from about 15 torr to 40 torr, led to a tremendous increase in the fractional saturation for hemoglobin. This is a consequence of the sigmoidal shape of that curve. We'll talk more about that next time. But please become familiar with how to read these beyond just simply looking at them with respect to KD. Try to make predictions about, if you are given the information, about the fractional saturations under different concentrations of ligand. Try to understand how fractional saturation varies with changing ligand concentration. Now, I always like to throw this out, though I think this will work a lot less effectively in recorded lecture relative to in person, but I want you to look at this plot and tell me, just by looking at it, which enzyme binds ligand A more tightly, enzyme X or enzyme Y. So as I'm looking at this, I see that they are both rectangular hyperbolas, so they are both hyperbolic in nature, which suggests that there's not any cooperativity that drives ligand binding. Okay, so if I just look at the 50% saturation mark, where fractional saturation equals 0.5, if I extrapolate over for X, so move on over to X, then extrapolate down to the X-axis, I have a KD there of about 2 micromolar for X. If I move over to Y, you'll see that number increases to about 6 micromolar. Okay, so if you remember, the smaller the KD, the stronger the interaction, the tighter binding that is occurring. So in this case, enzyme X binds ligand A more tightly, stronger, with a stronger affinity than enzyme Y. 2 micromolar is smaller than 6. So there I can say enzyme X binds A more tightly than enzyme Y. Okay, so now we have covered the first half of Chapter 5. We have discussed the basic foundations of ligand binding through a quantitative lens. We have talked about really an introduction to myoglobin. Next time, we're going to build into the concept of allostery, which is a broader description of cooperativity. And we're going to be there focusing on hemoglobin as a model system. There you'll see a lot more structural discussion, and we're going to then sort of explore different models for allostery, and hopefully we will cover it all next time.